All right, hello again. I'm excited to be able to share a little bit of work that we've been pursuing in terms of biomarkers and modulation strategies in LGS. And I just want to highlight that I'm framing this discussion with examples from my lab and also work that Drs. Lepore and Schrei have done, also at UCI Chalk. And Dr. Schrei is here in the audience, so if there are any tough questions, send them directly to him. And with that, let's get started. So in terms of the biomarkers that I want to focus on today, I'll be talking mostly about electrophysiology. And so I just wanted to do a brief review about what we know about how information is processed in the brain, because that really frames what a biomarker is in a relevant sense. And a key point that has been identified is that the brain is really simultaneously performing processing at multiple scales. And we have to be cognizant of that when we're thinking about biomarkers. And so the first scale is that of action potentials. And so these are the means by which individual neurons communicate with each other. And I have an example of an action potential waveform up there at the top. And these are functioning on a millisecond time scale. And you can really only see these if you're recording neurons within about a few hundred micrometers from the individual neuron. And so that makes it not very practical for recording in functioning brains for the most part in a large scale. But helpfully, the brain is also functioning at a larger spatial and a longer temporal scale, and this is what we call the local field potential, or the LFP is the research terminology. And this really means kind of the ongoing synaptic activity, so the population of neurons, what are they doing in general. And interestingly enough, in our brains and in rodent brains, this typically takes the form of oscillations. And so these are like fluctuations in the field, and they're going to be anywhere from once or so per second up to several hundred cycles per second or several hundred hertz. And these oscillations we found in basic science and also in disease states are very important because they're actually organizing the action potentials of neurons, and they establish communication channels between different brain regions. So why do we actually care about this, particularly as it pertains to LGS? Well, what's really interesting is that we have good evidence from animal models that these oscillations are more than just passive correlates of what's happening in the brain. They can actually be, in some cases, modifiable targets. And what they're doing is they're actually recruiting neural spiking patterns that are related to information processing. And so here I'm going to give you a really nice animal model example from the Zugaro group. And so in the hippocampus, there are these high frequency, which is a brain region very important for learning and memory, there are these high frequency oscillations known as ripples. And there's an example of what a ripple would look like if we were recording directly from the hippocampus in blue there. And these ripples have been shown to actually organize neurons that are recording information that's relevant to our ongoing experience. And during offline states, particularly non-rapid eye movement or non-REM sleep, these ripples are actually occurring very closely in time with another oscillation that we're all familiar with, the cortical sleep spindle. And what's been found is that this is opening up a window where the hippocampus can communicate information from to the cortex, and this is really important for mediating memory. But what's even more fascinating about this is that it's been shown if you're able to detect these hippocampal ripples, these high-frequency oscillations in real time, and you give the brain a zap of electricity, you do stimulation such that you evoke a spindle, so you're increasing the correlation in time between the ripples and the spindles, you can actually transform an unremembered experience into a long-term memory in a normal rat. So you have a biomarker that's actually helping you to enhance a brain function. And so ripples and spindles and their interaction actually seems to be what I call a driver biomarker, so for a key cognitive function, which in this case is memory. And so what about LGS? Are there electrophysiologic biomarkers that we could potentially use, not only to facilitate diagnosis, which of course we already do, but even as potential therapeutic targets? And so there's definitely a couple that we're already familiar with, and we've heard about these a lot. Generalized paroxysmal fast activity, or GPFA, interictal epileptiform discharges, or IEDs, and these IEDs are often going to come in repetitive bouts, which makes up the slow spike and wave, which is also part of the LGS electrodiagnostic criteria. And what's interesting is that we all know that these are really important electrophysiologic biomarkers for LGS, but how do we actually identify them on EEG? And of course, right now, we do this mostly visually, so epileptologists are looking at the records and identifying these patterns. And surprisingly, when epileptologists visually mark these waveforms, when multiple epileptologists look at the same record, and then they try to see how often they agree for individual waveforms, the overall agreement is actually moderate to low. So that's that inter-rater reliability there, and you can see for GPFA, it's really low. For slow spike and wave, it's better. And so this tells us that even though we know these biomarkers quite well, our ability to identify them reliably on an individual basis is actually not that great the way we do it right now. And it wouldn't really allow us to implement closed-loop interventions like the one I was talking about in the animal model based on this sort of detection. And so it would be nice if we could actually develop automated, objective approaches to develop these sort of biomarkers. And that's exactly what Drs. Lepore and Schrei have been working on. And they've been developing a new method for EEG biomarker discovery that they call S-EPIC, or scalp EEG pattern identification and categorization. And so basically what this is doing is you're identifying events of interest that are designated by high power in the EEG. So you can see an example of some of those bursts in the raw traces at the top. And then when you transform it into time frequency domain, you can see those really hot red colors, which indicate that there's high power there. And once you identify these events of interest, you can actually go through and extract a lot of their characteristics. How long are they happening? How are they spread across the brain? What are their key frequency patterns? And then you can put them into a clustering algorithm that actually groups events by similar features to try and figure out exactly what you have in the data. And so this method really replicated nicely our knowledge of LGS biomarkers. And so without actually inputting any specifications, this data-driven approach grouped events that epileptologists visually identified as GPFA into a cluster, so that's cluster 1 up there. And it also similarly identified slow spike and wave as a separate cluster, and that's cluster 2 there. And so this automated method can effectively detect our known LGS biomarkers, very promising moving forward. But of course, interesting question, did it detect anything else? And the answer was yes. There were actually 2 additional clusters identified in the data derived from the LGS patients. And these corresponded to these brief bursts in the gamma band, so like a higher frequency band, or a beta band, kind of a middle range frequency band, that were actually not identified as visually being an abnormal pattern by our epileptologists. And so this is suggesting to us that there's actually even more information present in this data that we're not gleaning right now by our visual identification. And these sorts of patterns could even be carrying key diagnostic or therapeutic information, and it really merits some further investigation. Okay. So what are the next steps? Of course, we need to really evaluate the performance of these kind of automated detection protocols for the established LGS biomarkers, and also understand what these new features on the EEG could actually represent. But a critical question, and a main question that I wanted to be able to address, is do any of these biomarkers actually relate to the disease source or to a cognitive function? Are they modifiable targets? Because if they did, then obviously they could be ways that we could use new therapeutic strategies. And so here we can really benefit from animal models where we can do a lot more in detail testing with our electrophysiology, with our behavior, and also with testing a lot of different neuromodulation protocols that just is not safe or possible in patients. And so for this, I'm going to highlight a bit of work from my lab where we're really trying to employ this approach. And for this work, the caveat, the disclaimer is that we use a rather generic focal epilepsy model to do this work. A lot of features are similar with LGS. We do use a progressive epilepsy model. We use a model that has a high burden of interictal discharges, but it's not an LGS-specific model. I'm going to try to convince you, though, that there are a lot of parallels and possibilities for adaptation, though. Another key feature of our model is that our animals do show progressive cognitive problems and memory deficits over time. And I'm just going to tell you a little bit about how we assess that in our animals. And so I'm showing you here a schematic of one of our animals' mazes. And so what we do is we have this open field maze that the animals explore. It has a lot of different hidden water wells. And of course, rodents are really good foragers. And so we train them that there is water in 3 locations on the maze, for example, where those blue dots are. And they learn it really quickly, and during the training phase, they will start making those very stereotyped red trajectories, and they'll retrieve the water right away. And at the beginning, before the animals have had a lot of seizures or interictal discharges, if you come back 24 hours later, they will do exactly the same trajectory. So they will tell you, I remember exactly where those rewards are that you trained me on the day before. And what happens is once the animals experience a lot of seizures and a lot of interictal discharges, they can still learn where the rewards are, and so that's the bottom left there. But then you come back 24 hours later and they don't remember. They're wandering around the maze, frantically trying to figure out where their rewards are. And so this is a key way that we can assess memory in our model. So we began our search for a cognitively relevant, potentially modifiable biomarker with the most common electrophysiologic epilepsy biomarker, which is the interictal epileptiform discharge, or IED. And so there's an example of one that comes from the hippocampus there in orange. And of course, these are these brief, spontaneous bursts of pathologic activity. They're not supposedly containing any interpretable information. So this is unlike the physiologic oscillations that we briefly mentioned, the ripples and the spindles. And of course, they've been associated with cognitive impairments in patients with epilepsy. So what do IEDs do in the brain, and can we modify it? And so work that we did was actually putting electrodes into multiple regions of the brain of freely moving rats, using our progressive epilepsy model, recording their IEDs, and looking for what happens. And what we found that was different is that IEDs actually really strongly couple with and induce reactions in other brain regions. And so in this case, the reaction that I'm showing is that they are able to induce spindles. So that may sound familiar because I was just telling you about the rats, how the ripples couple with the spindles, and that's important for memory. Well, in the epileptic brain, IEDs couple with spindles, and we think that this pathologic interaction is actually overriding the capacity for normal memory. And on the right there, what I'm showing you are just quantitative measures. And so the height of the peak shows you the strength of the coupling interaction. So for the ripples and spindles, it's significant but moderate. For the IEDs and spindles, it's really big. So we have a very strong interaction there. Of course, animals are animals. We always want to know if these findings translate to humans. And so we've already looked at this using intracranial EEG data from patients with refractory focal epilepsy who are undergoing workup for epilepsy surgery. And basically what we found is that this IED spindle coupling also occurs in the human brain. So I have some sample traces there and some of the same graphs that we use in our animal models. And in a nutshell, on the right, where we have that color map, what we can see is that there are certain areas of the brain represented by those warmer colors where the IEDs are really inducing a lot of spindles. So we have these pathologic windows of communication that are being opened in the brains of patients with epilepsy. So we think it's relevant for humans. Is it functionally relevant in humans? The only way we've been able to address this so far is correlative, of course, but what we did is we looked at this IED spindle coupling in a cohort of pediatric patients with refractory focal epilepsy, and what we found is that the patients who have the most pathologic coupling also tend to have the most problems cognitively, and so you can see that as the coupling strength goes up there on the x-axis, the IQ tends to fall lower and lower. And interestingly, these patients also tend to have poor outcomes after epilepsy surgery. And so even though this is correlational, it suggests that the coupling phenomenon could be contributing to functional impairments in patients. So, what can we do about it? So like I said, our hypothesis is that the IEDs are inducing spindles, they're opening up this pathologic communication, and this could be mediating cognitive dysfunction. Can we get in there, break this pathologic communication by using a neuromodulation strategy? So targeting this biomarker to see if we can get a beneficial effect on disease outcome. And so the way we did this was using closed-loop electrical stimulation, which will be familiar to you because that's exactly what responsive neurostimulation, or RNS, is. But in this case, what we're doing is targeting things a little bit differently. RNS, of course, targets seizures. Here, what we're doing is we're targeting interictal discharges. So we have a little device that we implant in our animals. We're recording their interictal discharges, and we have a processor that detects them in real time. Whenever we have an interictal discharge, we're feeding back stimulation to other parts of their brain. And we did a lot of exploration in animal models to try and understand which sorts of stimulation protocols are most effective for doing what we want to do, and we figured that out. I won't get into the details. But essentially, after all of this work, what we were able to do is employ this closed-loop stimulation in our animal models, where we detect the interictal discharge, we give the stimulation to the brain, and instead of having our pathologic communication in our coupled spindle, we have this period of relative silence. You can see that red line is pretty flat there, and instead of that kind of burst of yellow that we have on the left, you can see things look very blue. So we've kind of silence that pathologic communication. What happens when we actually apply this chronically, and of course we're doing this in an animal model, but what we were very surprised to see was that we had benefits not only on seizures, but also on the spread of the epileptic network. So here we have 3 groups of rats. We have one group which is in red, so these are the rats that just have the progressive epilepsy. They don't have any intervention. We have the yellow-orange group where they have kind of open loop stimulation and also progressive epilepsy, and then in green we have the closed loop, which they have progressive epilepsy exactly the same as the other groups, but they're getting our closed loop stimulation protocol. And what I'm showing there at the top is this kind of survival curve for focal seizures progressing to bilateral tonic-clonic seizures, and you can see that in the progressive epilepsy red group, that progression is pretty fast over a couple of weeks. We're actually able to stave that off quite substantially with our closed-loop electrical stimulation. And if we look at the electrophysiologic markers, we actually see that there are a lot fewer interictal discharges in our closed-loop group. And so this suggests that this type of chronic intervention is actually preventing the expansion of the epileptic network and the worsening of seizures. Of course, the main thing that we wanted to address in parallel to this is what's happening with the comorbidities. And so we tested the animals' memory throughout this closed-loop protocol. And what we found is that, of course, at the beginning, all of our groups have 100% performance in memory because they haven't experienced seizures and interictal discharges. And then as the epilepsy kicks off, you can see that for our red group, which is the only epilepsy group, their performance really, really decreases. The same with the open loop stimulation, but with the closed loop stimulation, we're actually able to preserve their memory. And so we are hopeful that this indicates some sort of ability to modify disease course. And of course, we're excited about this in general, but how does it apply to LGS? And I think there's several avenues of exploration that could apply. And so specifically, our closed-loop protocol is going to act by counteracting this, this slow wave that happens after the interictal discharge. That slow wave is present in LGS. We think that we may have the ability to prevent further cortical recruitment into the epileptic network, just like we did in this focal epilepsy model. And I'm not showing any of the data here, but we also do some work in the lab of regarding restoration of physiologic sleep rhythms, and we think that that could potentially apply to LGS as well. And we're excited to actually target GPFA and maybe even these novel beta-gamma epochs. And of course, one last point that we've already heard a lot about is how do we identify patients at risk and intervene early? And so this, again, is work from Dr. Schrey and Lepore, and they've actually found that functional connectivity, so that is the degree that the EEG is correlated over space, potentially predicts LGS diagnosis. And so these are some patients here, 3 patients that all received a diagnosis of infantile spasms, and that's chronologically there on the left, and you can see all those warm colors indicating high functional connectivity. And then when their infantile spasms were effectively treated, in the top 2 patients, the connectivity goes down. And then when LGS is diagnosed, you can see those warm colors start to come back. And interestingly, for the 3rd patient, that bottom row there, you can see that the warm colors start to come back even before the LGS diagnosis. And so this is a really interesting avenue of exploration for how we could identify patients and initiate treatments in the early stages, again, using these sorts of EEG biomarkers. So in summary, I hope I've shown you that oscillations can be targeted to modify disease progression and improve physiologic functions of the brain, at least in animal models. We need automated methods to reliably detect these sorts of oscillations and understand how they are expressed across the LGS population. We can use animal models to test causal relationships with disease progression and cognitive function, and we can explore neuromodulation protocols more freely. And I think that neuromodulation strategies can potentially hold promise for altering disease course, especially if we could identify patients at risk early. And so with that, I'd just like to acknowledge my group and collaborators and funding agencies, as well as our fantastic team at UCI and CHOC, and all the great work from Drs. Schrey and Lepore. And I think I've been a little bit over time, but if there is time for questions, I'm happy to take any. Thank you so much for your attention.
Beautiful talk, thank you. So you mentioned the IEDs. You kind of like touched a little bit at the very end, but the IEDs is one of the epileptic forms discharges. So do you think this could be applicable to other kind of like kinds of discharges in the brain? And kind of like related with that, in RNS, this is kind of like a naive question. In RNS for humans, right? So what kind of discharges are kind of like the red flag, that will indicate that you have to receive an stimulation, thinking on what you could do with humans in the future, right?
Yeah, exactly. So to the first point, the brain seems to have a lot of default or conserved pathways that it narrows towards in response to different forms of dysfunction. And so I think that a lot of the pathologic discharges that we see in the brain have similar underlying neural correlates. We often have excess neural spiking, hypersynchronous neural spiking. And so a lot of our strategies could really be tuned to identifying exactly what's happening at the neural spiking level and then just tuning our strategy to target that. So I think it's generalizable in a lot of ways. For the RNS, typically right now how the detection protocols are targeted is that they're looking for the onset of the seizure pattern, so an ictal rhythm. And so oftentimes that would look like low amplitude, high frequency, fast activity if we're really close to the seizure onset. And so once they pick that signature up in frequency and in time, then usually high frequency stimulation would be administered to the seizure focus to try and stop that from spreading. So that's what we do right now with RNS. We know that RNS stimulates the brain for a lot of things that probably aren't full seizure patterns. So we're probably doing a lot of interictal stimulation in RNS already. But I think what some of this animal model work is showing is that if you're able to be more precise and you're able to deliver stimulation protocols that are maybe more used to what the brain is used to receiving rather than these high frequency pulses, you might be able to have more enduring and also more comprehensive effects on the system. Yeah. Okay, I think we have to move along. So I'm happy to talk to everyone in the break, but I really want to give our next speaker the chance to chat. Thank you.